I'm going to talk about vaccine vigilance towards strengthening pharmacovigilance. He's already given you my introduction, but let's go on to the topic. There were 13 vaccines developed this century, meaning from 2000 till 2050. 13 new vaccines have come out in the market. Immunization is the most cost-effective and widely used public health intervention. A large number of vaccines are available. 37 shots to be precise for a child aged 0 to 6 years. They receive 37 shots of vaccines at the moment. And the incidence of vaccine preventable diseases decreases when the vaccine coverage increases, and when this disease incidence decreases, the adverse events due to vaccines and others, some coincidental effects, they become increasingly frequent and prominent. How do vaccines differ from drugs? Now, drugs are generally given to treat an illness or a syndrome, while vaccines are for prevention of illness. So it's basically intervention versus prevention. Adverse events to vaccines indicate immune response. They're an indicator of either a B cell response or a T cell response, and production of antibodies which protects the individual against the disease. Whereas drug-related adverse events are undesirable effects. They usually result, there's some form of organ toxicity in the affected organ. Now this affects the vaccines. They affect the entire analysis, interpretation, and implications of vigilance data. It is not the same as that for a tablet. Say you give acetaminophen or you give aspirin, you know what to expect, gastric toxicity and all. In this case, there's a wide range of adverse events. Now, vaccines are generally given in large campaigns. They're given in thousands of doses. I remember I was in Iran many years ago, and they had a campaign in which they gave 31 million doses of measles rubella vaccine to the whole of the country, to people aged between 15 to 30 years. They did not bother to check whether the ladies had an early pregnancy. Some of the ladies did not know that they had a pregnancy, and they received a measles rubella vaccine. So post-vaccination in that campaign, the WHO decided to go in and see after a year or so as to what was the effect of this rubella vaccine on fetal outcome. So about 2,500 samples were taken. They were analyzed. The ladies were followed up in their respective homes, because in Iran it was a decree by the Ayatollah. The coverage was 100%. So in vaccines, you really don't know, as to you don't have access to the provider who's given you the vaccine. Because the campaign is over, the people who administered the vaccine have gone away. So if a person has an adverse event, he doesn't know really where to go to. And even if he goes to someone, that particular person doesn't have your history or your records, medical records. So that's usually what I'm talking about, is more for a developing country than for the developed world, where everything is available in a database for each patient with his GP. So the drug AE results in a healthcare provider being informed to follow up and treat, whereas little information may be available on how many doses have been administered for a vaccine, to whom, and with what results. These are some of the challenges. There is no detailed safety information on vaccines prior to licensure, because phase 1 trials are generally on 50 to 100 patients. You observe them for 30 days, and then you say it's safe. Then you go to phase 2. Phase 2, you may have 100, 200, 300 patients, not more than that, subjects. And then clinical trials on immunogenicity and reactogenicity, because the blood sampling involved, you have about 300. The WHO says if you have about 300 patients per vaccine, means a comparator vaccine or a placebo trial, then you can have the vaccine licensed. Now, when the vaccine is licensed, it is used in millions of subjects post-licensure. And then in these clinical trial evaluations, the subjects who are at extremes of age are excluded. The children are excluded. The old who get a lot of vaccines are excluded. So you really don't know what the vaccine is going to do in that particular age group. And then the new vaccine does not have data on special groups like pregnant women, HIV, immunosuppressed individuals, cancer patients, and the elderly. You have the Global Advisory Committee on Vaccine Safety, which was established by the WHO in Europe to address vaccine safety in Europe. It stressed upon prompt data transmission by member states and assurance of the data quality and timely signal detection and action. It also stressed on safety of preservatives and non-antigenic ingredients in the vaccine manufacture and formulation. This point is very important because now we have vaccines which are coming out of, most of the vaccines are from the capsular polysaccharide of the organism, so they're made from that. But now you have vaccines which are being made by the recombinant DNA technique. You have vaccines coming out from common proteins inside the cell wall, and you have all kinds of vaccines. I'll be coming on to those, some very new developments. And so the adjuvants have become very important. Adjuvants are added to killed vaccines or inactivated vaccines to make them more immunologically strong to protect the individual, but they also have adverse events associated. So adjuvants like aluminium hydroxide, aluminium phosphate, stabilizers like gelatin, residuals like formaldehyde, viral growth media, vectors, all these porcine elements which are used during the production process, they all contribute to vaccine reactions. There you have the EUVAX project and the VENIS, the Vaccine European New Integrated Collaboration Effort, which is monitoring all these adverse events across Europe. Also you have the Uppsala Monitoring Center in Sweden. It receives vaccine adverse event reports as well as pharmaceutical product reports from about 100 countries across the world, and it reinforces the adverse event monitoring to vaccines. It is imperative that effective vaccinovigilance systems are developed, maintained effectively. In European Union, adverse events of special interest to vaccines have been classified as they may be linked to potentially serious adverse events. Now these have been AESIs of special interest are basically neuritis, convulsions, syncope, vasculitis, encephalitis, thrombocytopenia, Bell's palsy, and Guillain-Barré syndrome, which is acute ascending polyneuritis, a transient polyneuritis, difficulty in walking, problems like that. It's known to occur with influenza vaccines. There have been gaps in our knowledge on vaccine safety basically. There is inadequate scientific evidence to establish a causal link with a vaccine. You can only establish a causal link with the vaccine. Say for example, you have a measles, mumps, rubella vaccine. It's a live vaccine. So say you get aseptic meningitis to the mumps strain, you have to demonstrate the mumps strain in the CSF to say that fine, the causal link is established. That is not always possible. Some places a person may not be able to do a lumbar puncture. Then a laboratory may not be available to culture that possible particular virus. So the biological mechanisms of adverse events are also not very clear. There is insufficient or inconsistent information from case reports. There is an inadequate size or length of follow-up of many populations. A classical example in this length of follow-up is the HPV vaccine, Cervarix, human papillomavirus vaccine. When it was introduced, we didn't have an idea on what kind of protection, for how long it protected the ladies from human papillomavirus 16 and 18, which are known to cause 70% of cervical cancers. So now it has become possible, the studies were done, and they know now very well that Cervarix can give a protection from during that age, basically from sexual debut to say adolescence. It protects for about 6.5 years, and Gardasil, the Merck vaccine, the quadrivalent vaccine, protects for about 8 to 8.5 years. they've done studies to prove that. So there are limitations in existing surveillance systems also to prove causation. In developing countries, they still use paper. Adverse events following immunization, received from all the districts, from the periphery into a cell, say in my country in Delhi, and then they are transmitted further to the WHO. But the people who fill these forms, some of them may not even be doctors, some of them may be just nurses doubling up for the doctor. So basically the reliability factor is missing, and that becomes very vital when a reaction occurs. There's been a lack of expertise in pharmacoepidemiology, now evolved, and rare disease epidemiology with its special sets of methodological changes. Interest and resource allocation for vaccine safety is severely handicapped by the narrow negative terms of AE research when competing against the positive benefits and efficacy of a vaccine. Nobody wants to invest money in something which is going to be negative. That is what it was. Now things are changing because regulators are becoming very tough. Now things are changing. Even in developing countries, they have to do it the proper way. But earlier, as I said, the physicians, they didn't want to, they just wouldn't be bothered if you tell them that, please record the local and, the systemic reactions and all. They would say, we don't have the time. But now you have to, things are changing. Vaccine safety cannot be studied directly. It can only be inferred by the absence of adverse drug reactions when appropriate surveillance and risk management systems are used. Now, a balancing act is required in vaccine adverse event monitoring. This approach requires a systematic accumulation of negative findings, which may be more difficult to prove than positive findings. With the advent of reporting systems like the vaccine adverse event reporting system in US and the AEFI from WHO, and I think you have Vigilance over here, it is important for all national programs to implement AE surveillance for immunization. It has happened in our country also, in India, in developing countries. They have a national pharmacovigilance protocol, and now the regulators are asking for more and more company core data information sheet. They're asking for internal registry data. They're asking for all kinds of reports from all over the world before granting licensure to a vaccine. Recommendations for use of vaccines represent a dynamic balancing act of risk and benefits in which vaccine safety monitoring is essential. Because vaccines go to either the children and now they start going to the adolescents and to the adults also. Further research in vaccine safety can help distinguish true vaccine reactions from coincidental events and estimate the attributable risk as well as identify risk factors. There's a very good example in this was the interception which happened with the rotavirus vaccine. We will come to that. Then, the most sensitive part of a population receives vaccines. A higher standard of safety is required than other pharmaceutical products, which are curative. And tolerance of vaccines given to healthy infants is lower than the products given to sick people. This requires investigation into the possible causes of rare adverse events as compared to other pharmaceutical products. Then studies of rare events are less likely to provide definite conclusions, but they do give you a signal that this adverse event is occurring more than it should have occurred in a certain population, and an attributable risk of 1 per 10 raised to the power 5 is on the margin, 100,000 is on the margin epidemiologically. I remember doing a study in Egypt, all across Egypt, because they said that the MMR vaccine causes aseptic meningitis. It was reported from Brazil. This was the Leningrad Zagreb strain. Now, the incidence was mentioned as 1 in 100,000. So I had to organize the study with my team, and we did about 426,000 subjects. It's one of the largest trials ever done for safety. And we did not get any case of aseptic meningitis. We got one normal meningitis related to Haemophilus B influenza. So you require such huge samples to do these studies. or even for the rotavirus vaccine, they said Rotatech when it was withdrawn, 1 in 10,000 had the interception, and then the vaccine was withdrawn because they said, okay, this is proving causality. And then subsequently, when it was reintroduced, both the companies, Merck as well as GSK, they had to do studies on 60,000 subjects to prove or disprove the association with interception. Now, vaccines cannot be substituted. They have a very narrow margin for error. And an erroneous association can undermine confidence in a vaccine and affect the national program adversely. I've seen this happen on many occasions when there have been deaths. It has happened. So, it has happened in the UK also with pertussis, with MMR, autism, when the data was fixed. Later on, they came to know it was fixed. But for those 10 years, the industry was just talking about autism, nothing else. Finally, thankfully, it came out to be, it was not true. And parents started bringing their children back. Because when they don't get their children immunized, when the percentage of the population becomes more susceptible, then an outbreak takes place. A recent example was the pertussis outbreak in Australia, Canada, America, the outbreak of measles in Disney World, which happened recently. It happened because of this reason. Now, I don't think I'll go through all this. You know about the sample sizes for phase 1 to phase 3 trials. I'll move on. I have a lot to talk. Yes. Observations of common and systemic reactions are possible, whether done in comparison with placebo or double-blinded. However, there is one vaccine in which you cannot double-blind. That's the BCG vaccine, because it gives a scar on your arm. So you can't double-blind in that one. Better standardization of safety evaluations in phase 3 trials are required so that safety data across trials and vaccines can be compared. It is all about harmonization of clinical trial protocols basically across the world. So it is better if you do a multi-country, multi-center clinical trial. You'll get better answers. You'll be able to pool the data. You can have a meta-analysis. You can do anything. Now, the diphtheria, tetanus, pertussis vaccine, whole cell, has been, it's given to children who are 6 to 14 weeks of age or 2, 3, and 4 months of age in the UK. There's been a lot of debate comparing the whole cell with the acellular pertussis vaccine. The whole cell vaccine contains 3,000 proteins. It is very strongly immunogenic. Acellular pertussis contains either 3 proteins, PT, FHA, or it contains 5 proteins, PT, FHA, pertactin, fimbriae 1 and 2. So the acellular was compared against the whole cell DTP to look out for the local reactions, which are in the range of 40 to 50% with DTP, whole cell. And however, the definitions of high fever were variable, 39.5 to 40.5 degrees, oral versus rectal, where there's a difference of 0.5 degrees, and time after vaccination, 48 versus 72 hours. To overcome all this, the Brighton collaboration group introduced the Brighton classification for adverse events to DTP, and now you measure how much is the swelling, you measure how much is the induration, and you get some better answers. Major differences in the rate of hypotonic, hyporesponsive episodes with pertussis vaccine. Part of the DTP vaccine is pertussis. There was a difference in the Swedish and Italian trials, brought out the difficulty of standardizing assessment of rarer adverse events across cultures and health systems. Better assessment of vaccine safety before licensing may be needed. And fundamental to preventing safety problems is ensuring that vaccines are made under GMP with pre-release lot testing for safety and potency, preferably parallel to clinical trials before licensure. It is it's a very sensitive thing, these clinical trials. If the potency, there's a range. The WHO gives a range for potency of every vaccine. If you add a little more of one component, it reflects in the reactions, in the local reactions increase. And so you need a collaboration with the production department when you're doing these trials, when they give you the trial lots. between lot comparisons must be done before the clinical trial lot is assigned and given to you for trials. Post-licensing surveillance, it's going on in a big way these days. Bread and butter for some CROs and others, and they're doing a good job. You don't get the delayed reactions during the clinical trials, and these appear only later. There have been several instances like the nasal influenza vaccine causing Guillain-Barré syndrome, and then of course delayed onset was interception as well, was a delayed onset reaction. And then you have newer vaccines which have come in, the pandemic influenza vaccine, which has established a causal link with the Guillain-Barré syndrome, and many more. But it's all passive surveillance. All this is passive surveillance. So there have been some path-breaking methods from the statistical viewpoint and from mathematical modeling, whereby these issues are also being addressed. But at the same time, we have to keep doing a good job of it. You can do observational studies. You can do these observational studies, and they pose methodological difficulties. They are very expensive. You don't get that many number of subjects to be able to reach a conclusion. They are prone to ascertainment bias, and it is difficult to control individuals who do not receive a vaccine due to a contraindication or low socioeconomic status. Such individuals may have a different risk for an ADR than vaccinated individuals. And seizures or sudden infant death syndrome is also more common in the unvaccinated. In developing countries, there is an increasing recognition. In fact, most of the time I've attended several complaints the world over, but in developing countries, the WHO does recognize that most of the adverse events are due to program failure or a programmatic error, to put it in a better way. I remember visiting a hospital where one-day-old kids had been given 3 vaccines, which are given at the time of birth: BCG vaccine, hepatitis B vaccine, and there was another vaccine which was given. And the children died. 3 children died. And those 3 children had received 3 vaccines from different manufacturers. I was representing one of the companies. So by process of exclusion and all, whatever it was, hepatitis B is one of the safest vaccines you have in the world today. It was one of the vaccines which was received. BCG was received. So finally, it was my, after doing that investigation at my own level, representing the manufacturer, I concluded that the nurse had given this vaccine inside the blood vessel. Now that lady was not willing to accept that, and the pediatrician under whom she worked was not willing to expose her. So there was a big investigation conducted by the DCGI, and finally the nurse did admit that when she had withdrawn the plunger, blood had come into it, and so inadvertently she was responsible for the death of 3 children. Reconstitution with the wrong diluent. There's a guy in India who broke the vial of the diluent and then mixed it with tap water and gave it to a kid, had convulsions immediately. And toxic shock syndrome with measles vaccine. You're supposed to use measles vaccine within 4 to 6 hours of diluting it. In Nepal, there was a guy who asked one of his relatives to double up for him on duty, a health worker, and taught him how to give the measles vaccine. But he didn't tell him that don't use reconstituted vaccine beyond 4 hours. That guy used reconstituted vaccine the next day. 3 kids died. And Staphylococcus aureus was demonstrated in the needles, in the tubes. It was proven. Contaminated needles, syringes, switch off strains between vaccines, shots to Leningrad, Zagreb strain, problems with intradermal techniques, vaccine failure. People don't give intradermal injection properly. They don't ensure that there's a bleb of 0.7 millimeters. They're going deep into the muscle. This is one of the problems with rabies vaccine. Rabies is a 100% fatal disease. But people don't know how to give intradermal properly. So the Indian government did a pretty wise thing, they used the BCG inoculators who were well trained to give intradermal for giving this particular vaccine and rabies vaccine. But by and large, people are using it by the intramuscular route. We need constant vigilance. And then the child is with the mother. They're given a diary card. The baby is next to the mother. If he gets fever, at least she can record that my child was warm last night and got a fever. Most of our systems are passive surveillance systems, but they are the cornerstone of vaccine safety monitoring. They are low cost, and systems are in place for all vaccines in various countries in the world. In many developing countries as well, we have systems now in Canada, Denmark, India, USA, of course, leading the pack there. These are some of the vaccine reporting systems. I'll be discussing about VAERS from USA in detail because it's a really good, it's probably the best one at the moment. Then of course we have the adverse events following immunization, the Canadian system, then the yellow card reporting system in UK, of course, and the Aberdeen Dundee monitoring system, prescription event monitoring system, Indian Council of Medical Research, New Delhi, and Uppsala in Sweden. Apart from this, we have new systems as well. Now, I want to talk about this because it's quite fascinating. Vaccine adverse event reporting system was established in America in 1988, and then it was supposed to comply with the National Childhood Vaccine Injury Act of '86. It became operational on the 1st of November 1990, and all throughout the US, it was a joint CDC-FDA initiative. Initially, they said report the following events for DTP and polio vaccines with the time periods in brackets: anaphylaxis within 24 hours, encephalopathy within 7 days, hyporesponsive hypotonic episodes or shock within 7 days, seizures, death, no time limit. Then with MMR vaccine, anaphylaxis within 24 hours and encephalopathy within 15 days. These are live vaccines, so encephalopathy is more common. Residual seizure disorder within 15 days, or 3 days, or 2 seizures without fever or with fever, less than 102 degrees within 1 year of vaccine receipt. That is how the VAERS started. Now, all the vaccines possible which are licensed are included for reporting adverse events. Post-marketing surveillance is, vaccine lots are tested for potency, safety, and sterility, purity, identity, and all their constituents prior to the release. What happens is, say, I was working for Sanofi at some point of time. We had the FSAPs in Paris, which used to clear the lots which were coming to India. When they came to India, they went to the National Control Laboratory for testing, who would again retest the vaccine as per the protocols provided by the FSAPs, and then there would be a delay before, if it was found satisfactory, it will be released, otherwise the vaccine will not be released for general use. And then after that, there are methods that during phase 4 you do large post-marketing surveillance studies to monitor, especially for new vaccines, newly licensed vaccines, or new indications of a previous vaccine. Spontaneous reporting system is the one followed by most manufacturers in India, and it's a good system. It's the only one available at the moment. Now, Sentinel for detection of previously unreported vaccine AEs or unusual increase in reported events. VAERS will detect it. VAERS also has a lot of weaknesses, but the main positive influence it has is if there is increase of intussusception, if there's increase of Guillain-Barré syndrome, or any other or encephalopathy after certain injection, it will tell you how many people in America had this particular adverse event. That much it will tell you. And once the VAERS gives you a signal, then the vaccine safety data link will further investigate that particular adverse event and then reach a logical conclusion. So it gives a number of adverse events nationally. It examines the risk factors, provides vaccine specific AE data. It is relatively inexpensive but there is no laboratory or clinical data. Meaning thereby, you can't prove a causal association with VAERS, you'll just get a good signal about the adverse events. Weaknesses. How many number of doses have been distributed? Say you have a cluster of adverse events in 10 children. You go and see 2 different batches have been given. Then you go to the manufacturer and ask him, how many doses of this vaccine were distributed elsewhere in the world? Did anybody else report this reaction? But this kind of a data is not available to VAERS. It doesn't have a denominator. So, and there is no comparator group. So causality assessment is very, very limited. It's not possible actually with VAERS. It can just give you a good signal. Then there is underreporting into this database and inadequate information provided by the reporter, the same story. Passive surveillance, and of course sometimes what happens is that the media says that okay fine, like for autism when it came out, they said oh MMR causes autism. During those days you'll find more people reporting adverse events to MMR, over reporting, or you can call it like an overkill kind of a thing. So what they did was, they said, okay, let's have large linked databases. So they linked about 50,000 children on a large linked database. Post-licensure studies are expensive and test a limited hypothesis. Large linked databases are a promising approach for pharmacoepidemiological studies, derived from defined populations like universal healthcare systems. Information on exposure and outcomes is available. Underreporting and bias are minimized, and the denominator data is available. So the sudden infant death syndrome related with the diphtheria, tetanus, pertussis vaccine and seizures with MMR vaccine were discovered through these databases. So you have VAERS giving you a signal, and then the vaccine safety data link run by the CDC. It has 7 large health care organizations, the Kaiser Permanente and the very large organizations in the US. 500,000 cohorts are linked to conduct safety analysis based on reports and literature or concerns from VAERS, provide information to the Advisory Committee on Immunization Practices, which keep changing the recommendations and age groups every year. And what they have is active surveillance, rapid cycle analysis. Active surveillance is done every 4 weeks between a vaccinated and unvaccinated cohort to compare the AE profile. And they did so for Pentacell. Pentacell is a IPV, polio, inactivated polio containing pentavalent vaccine, registered by the name of Pentaxim in India, and Pentacell in the US, and I think Pentacell in the UK as well. So it's a DTP, IPV, and a Hib vaccine. Kindrix, rotavirus, and the AH1N1 pandemic vaccine, they were subjected to this rapid cycle analysis. So every 4 weeks, the main aim was to conduct a valid, accurate safety studies, a new tool to detect early signs of vaccine AEs, an increase in intussusception, which is a indentation of the intestine of the child leading to bleeding, was detected after 2,589 doses. About the same time when the reports of intussusception started coming to the VAERS for Rotatec, the Merck vaccine. Now this allows a rapid and routine population-based assessment of new vaccine safety, and Chen et al. And Davis, they've been working on it, and they have been using this. This is the vaccine adverse event form. Basically, I'll just tell you from my experience that the name of the vaccine manufacturer, the expiry date, the route of administration, the dose, the onset of the reaction, the temporal relationship, the time between giving the vaccine and having a reaction, and outcome and recovery from that, with sequelae or without sequelae. Those are very important points. Now the VAERS form, it gets inputs from 2 sides. It gets inputs from the manufacturers, and it also gets inputs from all the healthcare providers, nurses, everybody, even the parents can file a VERS form in the US. That is how you investigate a likely cause of cluster, whether it is program related or whether it is a coincidental event. That is why for intussusception, they have said that the course of all rotavirus vaccines should be completed before 14 weeks of age. Before 14 weeks of age, because after 14 weeks, intussusception per se naturally tends to occur more in the child. So they've kept certain limits in vaccinology based on that. These are the 4 elements. I'm sure you all know about these: adverse event or pharmacovigilance, identifiable source, a patient, a product, and adverse event or fatal outcome should be there. Minimum information. And this, of course, is the seriousness criteria, very important. Those adverse events which are fatal, life-threatening, or those which result in disability and prolonged hospitalization, congenital anomalies and births, and so on and so forth. These are the serious adverse events. What is the expectedness? An adverse reaction which is not specifically included as a suspected adverse effect in the summary of product characteristics. This includes any adverse reaction whose nature, severity, or outcome is inconsistent with information in the SPC. Company code data sheet, what exactly is it? Contains all the information related to the vaccine. And what exactly is vaccinovigilance? Is defined as the science and activities relating to detection, assessment, understanding, prevention, and communication of adverse events following immunization or any other vaccine or immunization related issue, that is catering for the adjuvants. Cluster, the number of cases reported after administration of the same vaccine coming from the same lot and from the same area and the same source over a short period of time. This was the Brighton collaboration group which has developed the definitions now, clear-cut definitions for fever, HHE, intussusception, nodule at injection site, abscess, swelling, pruritus, persistent crying, seizures. It's very difficult. I happened to go to a physician once who had injected 3 kids, he's supposed to give the injection here on the lateral aspect of the thigh. Not only had he given, and he was an MD pediatrics, I felt very sorry for him. He'd given the injection at the wrong place. All the 3 kids had fever with huge swellings, and the man was not willing to accept that he had, given a wrong injection. And representing the manufacturer, for me, was such a difficult thing, to tell him in a very, very firm but a diplomatic manner that you've made a mistake. It happens, you have to face it out. So causality assessment, basically, if it's a local reaction, it usually occurs within 24 to 48 hours at the site of the injection. It could be immediate or delayed temporal association. The temporal things are also defined: 7 days, 15 days. Is it laboratory findings? Any laboratory findings, like I gave you the example of MMR vaccine. Exclude other causes like any other disease or co-administered drugs, and what does the bibliography say? So overall, finally, you're able to reach somewhere. And these are the 5 types of reactions you get. Adverse vaccine reaction, which is normal, local and systemic, resolves. A trigger reaction. Trigger reaction is one in which, say, a child has a history of epilepsy, still given a vaccine, further aggravated. Programmatic error, most common in developing countries. An injection reaction or an anxiety reaction. There's a paper, a wonderful paper called as Mass Psychological Illness following MMR vaccine. I once went to Sri Lanka where the girls were being immunized with rubella vaccine, and one girl was being immunized inside the room and there were 4 girls fainting outside. So that was a psychological effect. That's what we're talking about, psychogenic illness. This is the SIOMS form. It kind of requires all the information which is like the VERS form. And then, of course, risk management plans. This has become very important set of pharmacovigilance activities and interventions designed to identify, characterize, prevent, or minimize risks related to medicinal products, including the assessment of effectiveness of those interventions. This is what it is. If they are more than 10%, they're very frequent. 1 to 10% are common. Infrequent are 0.1 to 1%. Rare are 0.01 to 0.1%. Very rare and exceedingly rare. These are some of the websites which are useful in knowing about vaccines. Of course, the Advisory Committee on Immunization Practices from the US publishes recent information and guidelines every year. This is a very fascinating case study, because as I was making my slides, there were so many things happening. But this one, I'll just tell you in a very brief manner that they reported after the pandemic, influenza pandemic, they reported in UK that there were children who were complaining of feeling sleepy in the daytime. That is what narcolepsy is. And they had received a ASO3 adjuvanted vaccine during the pandemics from GSK. Retrospect, this is open. I'm speaking openly because this is an open access article. Anybody can download it, anybody can read it. It was a retrospective analysis, a fantastic analysis done here by Elizabeth Miller, who I think is a huge amount of work on vaccines in the UK from the London School of Tropical Medicine. Whether it is measles, whether it's MMR, whether it's this one. So they did a retrospective analysis. Initially, the reports were denied by the manufacturers that narcolepsy can happen with the pandemic or the seasonal influenza vaccine. But in this case, clinical information and results of sleep tests were extracted from all the neonatal ICUs in UK between August 11 and February 12, and they were reviewed to confirm the diagnosis, vaccination, and clinical histories. So the sleep centers and the pediatric neurology centers in England did these studies in the age group 4 to 18 years in people who had onset of narcolepsy in the previous 1 year when they received the vaccines. And the odds of vaccination in those with narcolepsy compared with the age-matched English population after adjustment for clinical conditions that were indications for vaccination. The incidence of narcolepsy within 6 months of vaccination compared with incidence outside this period with a self-controlled case series method resulted in this. They had case notes for 245 children. Young people were reviewed. 75 of them had narcolepsy, 56 with cataplexy. Cataplexy is when narcolepsy is triggered by a surge of emotions. And 11 had been vaccinated before onset, 7 within 6 months. In those with a diagnosis by July 2011, odds ratio was 14.4 for vaccine at any time before onset and 16.2 for vaccination within 6 months. The relative incidence from the self-controlled cases, series analysis in those with a diagnosis was 9.9. Attributable risk was estimated as between 1 in 57,500 doses and 1 in 52,000 doses. These results proved a causal association between the pandemic influenza vaccine pandemics and narcolepsy, which was earlier being attributed to difference in genetics. That was mentioned in a very esteemed textbook. And they were saying, no, it cannot be possible. But when reports came from Finland with a similar attributable risk, even though more doses were used in Finland, causal association was proved beyond doubt and published in the BMJ. And this was the chart which they made: vaccinated before symptoms, unvaccinated, vaccination uptake, number of narcolepsy cases, and the population which was vaccinated. And you can see that a causal association was beautifully proved over here. The increased risk of narcolepsy after vaccination with ASO3 adjuvanted AH1N1 vaccine indicates a causal association consistent with findings from Finland. Because of variable delay in diagnosis, this risk might be overestimated, but EMA restricted the use of pandemics in subjects younger than 20 years. Seasonal influenza vaccines have not been linked to narcolepsy. Seasonal vaccines and pandemic vaccines are different. There are times when strains are common in northern and southern hemisphere. They can be similar also. But the seasonal influenza vaccine, like the flu shots you take every year, don't have that risk. Pandemic vaccine, they prove that risk. However, well, EMA just restricted the use of the vaccine in those below 20 years. And finally, as I said, adjuvants. The cause of narcolepsy may have been the ASO3 adjuvant, which contains squalene. Now, squalene is known to cause arthritis as well. It's an oil emulsion, alpha tocopherol, and polysorbate. These were thought to be the causes of this particular proven causal association. However, the vaccine is still licensed and being used in several countries worldwide. I think my time is going to be up in 10 more minutes, but I would like to tell you all of that wonderful development that we now have a phase 3 clinical trial completed on 7,500 subjects. And we have an Ebola vaccine, which is proven 100% efficacy using the ring cluster method in Guinea. And that is very good news for all the people in Africa who suffered from this illness and all those who even came to England and America. It's a very exciting development. At the same time, the dengue vaccine has not yet come out. The dengue vaccine is still being tried. CYD vaccine, it may come out very soon, but there was an issue with it that it protected the children during the first and second year. But after that, they found that the incidence of dengue was increasing in the third year. So further studies are underway, new vaccines coming out, really promising for the future. And I was extremely happy to read about this Ebola virus vaccine, because they had the Zaire strain of the Ebola virus. They put it in they tagged it on to the stomatitis virus, and the stomatitis virus on its surface was bearing the glycoprotein for Ebola. So the Ebola vaccine, the WHO, the whole world hailed this particular development. And I was being a vaccine man. I've been labeled a vaccine man even though I'm a physician and a pharmacologist, but I don't mind that. Even last night I was reading about it. I didn't want to load your brains with it, but I think telling you that much is enough. And so I think the future is bright, but we need to strengthen our pharmacovigilance systems for vaccines as well. Thank you very much for your patient listening. Thanks. And any questions, I'm happy to take any questions. Anything you want to ask me now or maybe later if you shorten down. Welcome.
A couple of questions regarding the case study.
Yes.
You said they restricted use in patients under 20 years. So is that the patients below 20 years of age wouldn't be allowed to use it? Why 20 years?
20 years because they studied it in 4 to 18 years.
Okay.
That's why.
And what, if any, was there any health impact on the population by restricting it in that particular age group? Because surely if you were vaccinating it beforehand, there must have been a reason for the vaccination.
Well, this was a pandemic vaccine.
Okay.
Unless the pandemic occurs again, there's no likelihood that a pandemic vaccine will be used. The routinely used vaccine will be the seasonal vaccine. But what happens is sometimes the strains of the pandemic vaccine and the seasonal vaccine, like the AH1N1 strain has become common. It is there in the seasonal vaccine also. So when the pandemic broke out, at that time the people who had received their seasonal influenza vaccine, especially the old people, they did not die. The maximum mortality was those between 40 to 50 who had not received a previous influenza vaccine. So I think a sound piece of advice is that please take your flu shot on time. It will save you a lot of botheration later. So I was associated with having the pandemic vaccine licensed in India during this pandemic. And I managed to do one of the fastest clinical studies ever done in the history of Sanofi Pasteur. In 2 months and 10 days, we licensed the vaccine, was licensed based on safety data, and 1.5 million doses were given to the health workers. But what happened? Half of them said, we don't want to take it due to the risk of adverse events. When the vaccine was not there, they were making noise. We don't have a vaccine. When the vaccine came, I think half of it was wasted away. So that's how vaccine programs need a lot of motivation and support, and only then can we have successful campaigns.
A lot of education.
A lot of education, health education.
Just one last comment, sorry.
Yes, sir. Most welcome.
I just noticed by just an observation that
Yeah.
in a case study done large. Very large there.
Yes. Yes. They have mentioned themselves that may have been an over-reporting or whatever. But the depth into which they went, examining the sleep records and the other things, and establishing the causal link. If they had not established the link, the EMA would not have revised its recommendation.
Absolutely. I'm just
Yeah,
saying
but definitely it had wide confidence intervals. Yeah, yes. But the whole lot of subjects, quite a large number of people received the vaccine, but it was lesser than the amount of vaccine given in Finland. And the results of this study reinforced the findings of the Finnish people that it was not by chance. Because everybody was saying it's due to genetic differences between the Finnish and the British and all that. No, nothing like that. Now causality was proved. And there's many more interesting observations coming up. It helped me quite a bit to do these slides that I was invited to write a chapter for Elsevier Academic Press on vaccines, which I was doing. So I was pretty well informed about my topic. So, and I thank you all for your patience and listening to me. I hope it was worth it. Thank you. Thank you very much.